Friends, welcome back to the new video, and in this video we are going to talk about very important topic again, that is the PCR, the polymerase chain reaction. And in this video we are going to learn who invented polymerase chain reaction, what are the steps involved in PCR, the advantages and disadvantages associated with the polymerase chain reaction, as well as the role of PCR in genetic engineering. So let's start the topic. Okay, so let's talk about the PCR. The PCR is invented by Carey-Mullis. Okay, he invented the PCR when in the year 1983. Okay, now it's a very versatile and revolutionary technique. Okay, so if we talk about the PCR, it is very versatile and revolutionary. Okay, and why this we will discuss in the when we are discussing the advantages. Okay, so it's a very technique. Okay, so why the PCR is used? Okay, so PCR is used to amplify the DNA segment, okay, of our interest through repeated cycle of DNA replication. Okay, and DNA replication, we all know that replication means copying. It means the parent strand is copied, okay, again and again in the repeated cycle. So now let's talk about the steps involved in PCR. Okay, steps of PCR. So basically there are 3 steps. Okay, the first one is I will just make it a 3 different step. Each will represent the one step. Okay, so the first step is denaturation. Okay, the second step is annealing, and the third step is elongation. Okay, now let's see what happened in each step. Okay, so first we will start with the denaturation. So in this denaturation step, the double stranded DNA, it is heated to high temperature, that is at 94 to 98 degrees Celsius. Okay, so double stranded DNA, okay, is heated at this temperature, and this temperature is it will break the hydrogen bond between the 2 strands and separating them to the single strand. Okay, so DNA, double strand DNA is converted to single strand. Okay, so in the optimal temperature for which is required for separating the double strand DNA is 94 to 98 degrees Celsius. Okay, now let's talk about the annealing. In the anneal, once the DNA strand, double strand is separated into single strand, then the temperature is lowered. Okay, so the temperature for annealing is around 50 to 68 degrees Celsius. Okay, so what will happen in the steps of annealing, there is a single stranded DNA molecule which we call primers. Okay, they will bind to the complementary sequence on the template strand. Okay, so the primers will bind template strand. Okay, okay, we should always keep in mind that the primers, they are designed to flank the specific DNA region that has to be amplified. Okay, so then the third step is the elongation. Okay, so in the elongation step, the temperature is raised at 72 degrees Celsius. Okay, and why? Because this is optimal temperature for DNA polymerase activity. Okay, the DNA polymerase activity. And DNA polymerase, what it will do, it will extend the primer. Okay, and it will extend the primer by adding the nucleotides to the 3' end to the synthesizing new DNA strand. Okay, so DNA polymerase will perform its activity and synthesize new strand by adding the nucleotide to the primer sequence. Okay, so you got my point. I will repeat it again, that when we start, there is a double strand DNA. Okay, just I will draw it like this. So this double strand DNA, it is in the denaturation step, it is converted into the single strand by heating at 94 to 98 degrees Celsius. And in the annealing step, what will happen, that to this we will design a single stranded nucleotide sequence that is known as primers. The primers will attach to the complementary sequence. Okay, and once they are attached in the annealing step, then the elongation step will come, and this is the primer. Okay, it has already attached during the annealing process. Then the at 72 degrees Celsius, the DNA polymerase, it will start adding the nucleotide. Okay, one by one. Okay, so by this way, a new DNA strand is produced. Now to this cycle of denaturation, annealing and inversion, it is repeated multiple times. Okay, around 25 to 35 times. Okay, this cycle is repeated, and it will give the billion or millions or billion copies. Okay, billion copy of targeted DNA. Okay, so that targeted DNA what we want to amplify will be amplified through the polymerase chain reaction. Okay, okay, now let's talk about the role of PCR in genetic engineering. So basically there are 4 applications that is of PCR in the genetic engineering. First one is the gene cloning. Okay, so in the gene cloning we will amplify specific sequence. Okay, of gene of interest. Okay, then that amplified gene, it is inserted into vector for manipulation and expression. Okay, so it is used for gene manipulation studies. Okay, the next application is the site directed mutagenesis. So okay, so in this site directed mutagenesis, the PCR, it is used to introduce specific mutation into DNA sequence. Okay, so into the in the DNA sequence, a mutation is introduced. Okay, at a specific site. Okay, now why it is used, like what is the benefit of site directed mutagenesis, that it allows the researcher to study the effect of mutation. Okay, the third application is in gene expression analysis. Okay, so by the PCR we can quantify the amount of mRNA produced from a specific gene under a specific condition. Okay, so it will provide insight into gene expression, so which genes are expressed. Okay, suppose if a plant is exposed to a cold stress, okay, then normally the normal plant will not have the genes express that will help them to cope with the cold stress. But when we put it into the cold condition, the gene related to the cold stress, they will start expressing. Okay, so that the plant will survive the cold temperature. Okay, so PCR is used for the gene expression analysis. And the last is diagnostic PCR. Okay, so diagnostic PCR, it is used to detect the presence of specific pathogen or mutation. Okay, causing a particular disease. Okay, so I hope you understand the role of PCR in genetic engineering. To give a quick glance again, the PCR is used for gene cloning. It is used for the site-directed mutagenesis. It is used for the expression of the gene. Okay, what genes are present, what genes are absent, and it is also used for the diagnostic purposes. Okay. Now let's talk about the advantages of PCR and disadvantages. Okay. So the first advantages, we will talk about the advantages. It is highly sensitive. It means that if even if we have like a very small trace amount of the DNA, it will amplify it. Okay. Specificity, if the design primers they are properly designed, then only the specific DNA sequence will be amplified. Speed also, it's like a very highly fast process, and it is completed within few hours. Okay. And versatility, and versatility means it is used for different kinds of application that we have discussed above. Okay, so these are the few advantages associated with the PCR. Okay, now let's talk about the limitation, so or the disadvantages. So it's highly sensitive to contamination. Okay, it means that if there is a slight contamination by a bacteria or anything, fungus, then the PCR will amplify that also. Okay, that we don't want actually. So it is not only highly sensitive to is a positive point, but it is highly sensitive to the contamination also. Okay. Seconds, the we have already discussed that it is if the primers are designed carefully, then it is very specific. However, if there is some problem in the primer designing, and it is not very specific, then the non-specific amplification will come. So we require the careful primer designing. Okay. And the last, but not the last, the second last, there are some PCR inhibitors also. Okay, so it is prone to inhibitors, so that inhibitor can affect the process of application. Okay, and last, there is sometimes the non-specific amplification and background amplifications also. Okay, we are trying to clone one gene of one family, but if there is quite similar there is a quite more similarity between the genes of that family, then if you want to clone suppose a gene, and there are 10 genes in a family, so they are highly homologous to each other, then if you want to clone a gene, but what will happen, sometimes the non-specific amplification will be there, like the part traces of the b or c genes will be there. Okay, so this is the limitation. But although the conclusion is that about the PCR, that it's a essential tool for it has emerged as an essential tool for molecular biologist. Okay, so now if we talk about the molecular biology, we cannot talk about the molecular biology without the PCR. Okay, so I hope I tried to make all the points clear to you. Thank you for watching the video.